I'm gonna write our rent check and put it on the breezeway.
Hello, hello. Jacob, can you hear me?
Yeah, I got you. Are you Max?
I am. Yeah. Hey, how's it going?
Awesome. It's going well. I'm trying to get like oriented here. I have to figure out how to convert you from an here we go. I'm promoting you to a panelist. Now you should be able to have like full control over your own stuff.
Okay, let's see here. Yeah, I can see. I can turn my video on.
I see you.
That's okay. Great.
Can you double check that you can share a screen? I might have to stop my share in order for that to happen, or maybe I don't even. I'm not sure.
You bet. Yeah, let me try. It says host disabled participant screen sharing.
All right, well, let me make sure that I enable that. I'm going to make you a co-host.
Okay.
How about now?
Still says host disabled participant screen sharing.
All right, I'm going to make you a co-host again.
Super co-host.
I would have thought that making you the co-host would enable you to share, but maybe let's see what happens if I stop my share. Oh, okay. I see. If I stop my share, then I see the dialog box that actually prompts me to make you the co-host.
Oh, very nice. Love it. It says I'm a co-host now.
Cool.
Cool. So now I can share my screen. Let me see if I can actually do that. Do you see like a web browser?
Yeah, I see a web browser with a Zoom something.
Slides, stuff like that.
Yeah, I see slides and stuff like that. Awesome.
Okay, perfect.
Well, we have about 80 registrants, which is pretty good, I think. We'll see how many of them actually show. Right now, we've got, and I think people can hear us. Like anyone on the meeting can hear us, by the way. Of which right now there's one. Hello, Ramiz. I know you can't talk to me, but I can say hi to you, and we'll see who trickles in.
Awesome.
All right, yeah. Era is gonna be here very shortly, I think. A little bit of confusion that might be more general, actually. So I'm gonna send another email to all of the attendees real quick. All right, I have some people are getting, ooh, Ara's here. I'm gonna make him, promote him to panelist. Sorry, not Ara, Era. And now I have to find where I had the thing that would let me make him a host so that he can present. Hey, Era, how's it going?
Oh, good. How are you doing?
I'm doing well. Sorry about that little bit of confusion there. I just fired off an email to all the attendees because I can't imagine you're the only one.
I was just being a little slow. I was like, oh, that's how you do that.
Cool. So just so you know, we have some attendees trickling in, and I think they can hear everything that we say. So we're on display at this point.
All right, I'll keep that in mind. So I should get my pets out of the way.
And great, both you and Max, actually, really great to meet you both. I think this is my first time like talking to either of you with words.
Oh yeah, that's kind of strange. That's right.
Yeah. Yeah. Hey, what's up? Yeah, thank you for organizing this. I know it's quite a bit of work, but I really appreciate it.
Yeah, well, I think assuming that that some more people trickle in, it should be a really good. Well, it'll be fun for us either way, I guess. But let's see. Then the other thing that I have to set up, actually, well, I can just do that later. Never mind. Cool. Era, I'm gonna still figure out how to navigate around Zoom enough to promote you. Got it, to a co-host. I don't think it's as relevant for you because I do have your slides that I can stream from here, but can't hurt, I guess.
Yeah, just in case something goes wrong, I should be able to try it on my end. It's a little weird on these Ubuntu machines with Zoom, plus our infrastructure here. Well, we have flash flood warnings for today, so it's going to be interesting here in a few minutes.
Oh, baby. All right, and Harold is now here.
Hello, can you hear me?
I can hear you, and I'm making you a co-host now, which I think will make the presenting a little smoother. Just so you know, Harold, oh, and Max is here too, so I'm gonna promote him as well. He's the SBC representative, Max Monte. So I'm gonna make him a co-host.
Hey.
Okay. And just so all of you know, we are all already live to some set of attendees, and so we're on display. Don't do anything too wildly embarrassing. And with that in mind, let me go ahead and share my screen back to this thing. And then I want to dump the registration link for part 2 into the chat here. So let me find that and do it. Hopefully we'll get all the bugs ironed out in part 1, and part 2 will just be a breeze. I have no idea what I'm doing if that's not clear. So
It's going great so far.
One thing, just to let you guys know, so it turns out after going around in circles with like help docs and whatever, we can't do breakout rooms in a webinar. So my solution, such as it is, since we're clocked in at just under 100 participants, is going to be to invite everyone after the main part of the meeting when it's time for the social, to move over to my personal Zoom room, which will be set up as a meeting rather than a webinar, so that we can socialize in breakout rooms there. So just so you know, that's coming, and no one is particularly obligated to move over there and show up, but we'll see how that goes. Let me track down still. I gotta find where I have this link. Not there. Here. Okay, now I have to find my way back to working my way back to you. Chat. Message, not just Max, but everyone. Link to registration. Right. German Max, do you want to, when the top of the hour rolls around, do you want to go first? Do you want me to go first and say a couple of words, or how do you want to organize that?
Yeah, I think it would be better if you would go first, and then I'll just say a couple of words, because yeah, I don't know exactly what's coming up, right? And are you speaking after me then again, or do
So,
we just
no,
straight
so
in?
I think well, so I'll say a few words welcoming people to the event, and then I will turn it over to you to say what you need to say, and then either we'll have a little bit of downtime before 12:15, or if we don't, I'll go ahead and introduce Era as our first real speaker.
Okay, cool. Yeah, sounds good.
Or at least I'll just say first we have Era, and I'll let him introduce himself.
Also
maybe
good.
maybe that's
For me
the way to
just.
do this. And I'm also pulling up a that's not what I want. Yeah, pulling up the actual schedule for this thing, so that I can consult it and know what it is. Cool. All right, well, I have that it is the top of the hour now on my end. We're still getting some people coming into the room at a fair clip. So I might hold off for 1 or 2 more minutes. Dump a notice to that effect in the chat so that people as they arrive know what's going on. Stop my screen share for just a sec to make sure that we get. Got it. Let's see. I'll share my screen again. This is the one I want to share. And okay, well, welcome everybody. We've still got people coming in at a pretty good clip, but there are more than 40 of us here, which is slightly more than half of the registration. I don't know if that's a good rate of return or a bad one, but seems like we've got a critical mass. So I guess I'd like to get started, make sure that we move on time here. I'm Jacob. Thanks so much, everyone, for coming, and thanks particularly to our speakers and panelists who are joining us today. In just a moment, I'm going to turn it over to Max Monte, who's going to introduce a little bit, just talk a little bit about Stan and this series of events. I'd like to make sure that everybody notices that in the chat, at the very top of the chat, and I'll put it in again once I'm done talking, because I think if you've joined afterwards, you might not be able to see it. But in the chat, there is a link to the registration for part 2 of this event, which is next week on October 4th, same time. And it's just a super great lineup of speakers that we have on both days. But I really encourage everybody, if they possibly can, to try to make it on October 4th. Thanks, Harold, for dumping that over. This pair of events was designed to be sort of a grab bag of what people are doing with Stan in ecology, at least as reflected in the people who answered the Discourse post. I'll mention that we are not so reflective of all of the great work that some people are doing with fish ecology or aquatic ecology in general with Stan. And that sort of is what it is. I'd also just like to sort of acknowledge up front that if we look across the 2 days of this event, we have a bit of a diversity shortcoming, and I want to accept real responsibility for that and apologize for it. And on the other hand, also acknowledge that it is a feature or a bug, I should say, in our intersecting communities of Stan users and statistical ecology. And so I hope that next year, hopefully we'll be doing this again, and hopefully we'll be able to have a more diverse group of speakers and panelists and so forth. So with that said, the sort of guiding principle behind these two events originally was that today we would be talking a bit more about sort of wildlife survey stats, and then next week we would talk a little bit more about all of the other crazy and wild and wonderful stuff that people are doing with Stan and ecology. And as things have shuffled around, and that distinction has broken down a little bit. But today we're going to hear two talks from Harald and from myself that are really about sort of core wildlife survey methods and methodology, both focused on occupancy modeling. And then we'll be bookended by presentations from Era and from Max, who will be talking about different parts of sort of the workflow sensu lato, that is all the way from how we engage stakeholders and build priors to how we implement our computational workflows on our computers. Raul says he can't hear me anymore. Can anybody else hear me?
You're
Okay.
back. Yep, we hear you.
Great. Okay, I apologize that my audio breaks up. Hopefully that won't be a persistent problem here, but we'll see how it goes. So with that, I'd like to turn it over to Max Monte. I hope I have your name approximately right, Max, and I'll stop my screen share and let Max say a few words.
Yeah, I call myself Max Montage, but yeah, it's a weird name, even in German, so it's fine. Yeah, hello, I'm Max. I'm part of the SGB, the Stan governing body. You can find about what we're doing here. I'll post a link in the chat. Just scroll down and you see sort of a list of the stuff that we are doing, we as a group. We are also responsible for StanCon. This year there's no StanCon, but we figured that we want to do something to foster the Stan community, and we came up with StanConnect. And because we saw that there are so many great sort of communities within the overarching Stan community, that we wanted to have sort of like a seminar series of people sharing the stuff that they are doing in Stan, because I think that this is the most exciting part about Stan, right? That we can sort of see applied stuff being done in Stan. There's actually, this is actually the second event in this series of events. You can find the other ones in the link that I just posted. And yeah, if you think that this is something that you want to do like in future, I think just write an email or post on Discourse, because we really want to get the whole community involved. I know we have a lot of ecology stuff being done in Stan, which is great, but also if you know other people in other fields, that would be great as well. Yeah, also, if you are in here, you probably know about the Stan forums on Discourse. I'll just share the link to anyone who doesn't know about the forum. It's a really great way to learn about Stan. Lots of great people answering questions about Stan in general, but also stats and different models. It's just a great place to be. And yeah, hopefully we will build the community even stronger, although we cannot meet in person like we would do in a typical StanCon. Yeah, and the last thing that I want to mention is that we actually want to have the community as inclusive as it can be, and have the barriers to entry as low as can be. So you probably know that StanCon is a pretty cheap conference to attend. Like if you disregard the travel costs. And also we want StanConnect to be free. That being said, or Stan as a project still relies on donations, right? So it would be really great if any one of you, or yeah, all of you would consider at least donating at the link that I just posted. We actually set the minimum amount of donations down a little bit, so just feel free to like donate the least amount, but every little bit helps, right? Yeah, if you have any questions or want to share something with the Stan governing body, you can also write us an email, the following email address. Yeah, hit us up with any questions you have, or any complaints you have, or yeah, if you have a suggestion or something, just write us an email. And yeah, that's it from my side. I want to say a huge thank you to Jacob and basically to everyone who organizes a StanConnect, right? And Jacob is like really, really amazing, and he organized 2, which is like incredible. So thank you, Jacob. And yeah, I'm really looking forward to all the talks and all the speakers and discussion. Yes, thank you.
Arrow, we're like 3 minutes ahead of schedule. I don't know if you have anyone who like you expect to be careening in right at 12:15, or if you'd rather take advantage of them and get cracking. I'll go ahead and share your slides and take it away whenever you want to.
Oh, great. Okay. If I had known that we wanted some fishery stuff, I could have brought in the fishery example that we're working on. So sorry about that. My name's Arrow Winter. I am what I always tell people, I'm an adorably queer New Mexican who just happens to be a sort of quantitative ecologist. I spend most of my time in arid land aquatic ecosystems. And I'm at the University of New Mexico in the Department of Biology and underneath the Bosque Ecosystem Monitoring Program. And I'll talk a little bit about that, but not too much. It sort of ties into the work here. If you go to the next slide. So in case you don't know, New Mexico is there in red. A lot of folks don't know where we're at. We are actually part of the United States of America. That can cause a lot of trouble when you're trying to get things shipped here sometimes. And it's a wide range of landscapes and ecosystems that you can kind of romp around in, and all in very short distances. So it's kind of a pretty cool place. Next slide, please. So specifically, we work in the middle Rio Grande Valley, and our science and education take place on current and forcibly stolen ancestral lands of the Pueblo, Dene, Apache, and other indigenous peoples. So we always need to keep that sort of front and center. We work with many sovereign nations as we're handling their data and a lot of their modeling questions. So it's important to take that first step in recognizing that the land that we're on was taken from them, particularly in the middle Rio Grande Valley here, all on the river. These points that you see here on the map, these are our research sites. We cover 350 kilometers of riparian zone, all up and down New Mexico to the far north to almost down to the border. And this is where I'll be talking about our stakeholders. And the talk is largely focused on how do you do model building and priors with stakeholders, particularly when every single research site is put in by request of a stakeholder. So they have different goals and agendas in what we do. So this talk is largely going to be in 2 pieces. There's sort of agendas and principles. If you're familiar with the Apocalypse World role-playing game, those are being pulled from there and slightly reworked for an academic setting. And then the latter part will actually be an example of a current conversation that's ongoing around modeling dragonfly behavior. Next slide, please. So model building is basically community building. Is you're usually sitting down with somebody across the table, or even at the same table, or a bunch of people, and you're basically coming up with a group environmental narrative. And so what I mean by that, and this is kind of like a shift in how we think about our models, is that an environmental narrative is what is happening right now, what has happened in this place, and what could potentially be happening. And using Stan and sort of the principal Bayesian workflow with that paper that came out, I believe that's been making its rounds on the discourse community, you can develop these transparent principal processes for getting at this underlying narrative and story. And it leads to a better mutual understanding between everybody. If I sit down at the table, I have a very narrow range of what I know and how I know things. But working with stakeholders who want us there in the first place, they give me a better understanding of the system and a sort of a different way of thinking about things. And this leads to buy-in. So a good example of buy-in are my two cats did not have a very good buy-in about who can share that top of the shelf. By sort of getting them together and maybe a little bit of catnip, which I don't recommend with your stakeholders, you can get buy-in. We now have a policy that says 2 cats can fit on the shelf. We'll be seeing lots of cats because I have a bunch. So they're part of the story. Next slide, please. So I often say, and sometimes I say we, but what I mean is I write a model and I'm making some often bold statements about statistics and mathematics in the model, right? But that statements don't always go over very well. People often want discourse, and you can see this discourse happening like at the Stan forums, right? There's talk around the model as we kind of start approaching consensus. And people were desperate for discourse, like the guy on the left. He really wanted to reach out and have some connection. And so if we start thinking about our models broadly more as an ongoing dialogue that is constantly evolving, this brings more stakeholders to the table. When you make statements, such as using the golf putting model on Stan about where that tennis ball should be and who owns it, statements lead to some conflict and unpleasant things happen, like this fight that was about to break out a couple of days ago. So think of them more as ongoing discourse and dialogue as opposed to statements that are set in stone. Next slide, please. So this may be hard to see at the moment. We'll get to a better version of it later on, but when you present a model, what story am I telling? And then did I listen? And so this means sort of being present by invite, and that not just the physical invite to come into a community, but also like thinking about what is the space, what's being demanded here. And we'll see this model, again, this is actually an occupancy model, a bunch of dragonflies, but we'll get to why this was absolutely incorrect. Next slide, please. All right, these questions came out of the Discourse forum, and I can't remember who said them originally. I went back and looked, but they circle around quite a bit. So what story am I telling here when we have y with the little tilde and 1, which is like essentially the intercept model? You notice it's not quite filled in, but when we show this model, what we're actually thinking is a step back, and we're asking, what does your data look like? What questions are you asking? What decisions are you going to be making? What inference are you looking for? So that's all actually bundled into this sort of very shorthand in that model statement there. Next slide, please. But often these 4 questions that are up here are more important. So draw up your models and leave blank spaces. So we're leaving a blank space in N. We're terrible blank spaces because we haven't put any priors in. We're talking about, I mean, I don't want to talk about why that's terrible, but we'll show what we do with that space. We want to know is who will be explaining the model? What do you personally as a stakeholder want from this? What does your manager want? If you're not the manager, they're going to be looking at this at some point in time. What do they want? And then what prior information do you have? Sometimes these will take more precedence than the first set of 4, which is ones that I routinely ask. These have been sort of developed as I've been talking with stakeholders in our ecosystem here. Next slide, please. So what do we, when we're talking about priors, we want to fill in those blank spaces. So this is, we can incorporate multiple stakeholders' knowledge. If there's like something really contentious parameter, we can actually model that as a prior, right? If somebody's like, well, this effect is negative, no, it's positive, no, this has absolutely no impact in the system whatsoever, we can actually work that in there and encode it into the Stan models. We could also make things like field notes, relic populations, and oral history if you're in ecology and walking around. Like over here is a field of yerba manza. This is very important. It says historical importance and cultural importance to us here in New Mexico. But it also tells us that there was a floodplain here at one point in time, even though it doesn't flood anymore. And that's information that you might want to know about. It's like when we had somebody's grandparents taking us around and they're like, yerba manza used to be in this area. They're like, oh, okay, so that means this was a floodplain. We can work all that kind of information into our priors. So I often present our priors as additional stories that we want to tell that are not necessarily driven by the data that we were collecting. Next slide, please. And how do we go doing this? Well, I like to bring everybody to the table. Oftentimes it's at my house. I can seat it around the table and I make really good food. And so we'll sit around and we'll draw probabilities. So we'll hand out index cards that have the different parameters on them, often coefficients. And we ask all stakeholders, draw your distribution. Like, do you think this is gonna be a positive effect, negative impact, or nothing, or does it include 0 in it? And so often we'll get a bunch of these index cards that are centered around different coefficients that we have, or even across, if we're thinking about hierarchical models, like what is that interaction among the different levels? And when I say, well, what do you think this should look like? And then we can start having discussions around how we know these things, why we know these things to be true, and then we can work them into the model. And this at least allows everybody to have their opinion heard, or rather seen in the shape of a distribution. And we can, scan these in and we keep them for a long time. And we start to build consensus around a coefficient, particularly if it's something that's doing with like an endangered species, that can often be really contentious. Next slide, please. So I do have a speech impediment. So there's certain words like the one in the upper left-hand corner I cannot say properly, and the things with L's in them I struggle with as well. So I can't pronounce that, but we're looking at dragonflies and damselflies, and we're working with the Pueblo of Santa Ana. So we're inside a sovereign nation carrying out some work. We got asked them to help. We have a long-standing relationship with this Pueblo, so it wasn't really, there was not a lot of complicated things for us to go through to actually be able to carry out this work. And we're not doing the survey, but we're wrangling the data and the modeling. All right, move to the next slide, please. So remember this? Okay, I talked about this. Now you can see it a little bit better. So I blew the species. This is an occupancy model. So I was sent the data. Of course, being an ecologist, I'm like, oh man, we can totally throw this in an occupancy model, because the stakeholder was like, I want to know something about richness. And I was like, okay, yeah, totally occupancy model. And then you send the Stan code back and it's a wall of Stan code. It's not that bad, but imagine you're a stakeholder who does R, but you don't write models from scratch, getting a wall of Stan code back, right? So it was like, oh, this is cool, but very academic. Right. So he's like, let's put this aside and let's talk about counts and richness, but not so much about the occupancy model. We'll save that for a paper that we're going to write. But this needs to go to my manager, who needs to explain it to the tribe about why we should do certain things, take certain management actions or not. So the question is like, who will be explaining the model? What do you want from it? What does your manager want? Actually drove the model totally away from the occupancy part and just into something else. So next slide, please. So we often talk about data, but when we're, particularly when we're working on the Pueblos and other communities, our conversations are around the data. We're out in the field a lot, and that's where the conversations happen. You'll be waist deep in a pit of water with a bunch of cattails and yerba manza around you, right? And then you'll be talking about what has happened to this landscape and what might be happening as we, enter into, climate change and higher variabilities. So here's actually what the data was. It was like, okay, there's 2 ponds we're looking at for dragonfly counts, doing them daily or several hours every day for 4 or 5 months, however long dragonflies live. I'm not a bugologist, so I can't answer the biology part of this thing. So this is what the data actually look like. So it's like, okay, so we have some different questions, not necessarily occupancy thing here, but there's stuff happening. So next slide. So this is where we can start with a model, just a simple intercept model. So this might be like, yeah, or you could have filled this in. You can't have negative dragonflies. But again, we're presenting a model to a stakeholder, and we start talking about why do we have all these parameters in here and what do they mean? How do we fill them in together? So putting a lower equals 0, which means we can't have naked dragonflies because that would be really weird. Starts to invite the stakeholder can laugh, I can laugh at myself, and then we can be like, okay, yeah, so let's start thinking about like the biology that we can encode into this model and the different ways of knowing about the data. Next slide, please. But what is actually happening here is not just wanting to know something about the mean of the richness between the two ponds. There's actually management going on. They're mowing around the ponds. And we're starting to wonder, does that have an impact on the dragonfly species? Because they are culturally important, but also they're important in riparian zones where we have very sort of intermittent water that comes through the system. So this is what we actually want, is basically an ANOVA. And so the testing and things like that come up. So this is where the second slide gets super important, or the slide after this one. So next slide, please. So if you hear the word testing and ANOVA and p-values, don't roll your eyes like this cat's doing right now, unless you are that adorable. Because otherwise, people are going to be like, yeah, you're crossing a line here, and then that shuts down conversation. So what we can focus on is, what is the purpose of testing? Do you want to quantify uncertainty, and what kind of uncertainty can you tolerate? And that shifts the conversation back to, oh, we can express a lot of these common statistical tests, which people will rattle off to you, in a Bayesian linear model, and it's often way more informative. And for me, it's way more fascinating as we start to dig out what the priors are. So next slide, please. And so we have this ongoing dialogue, and it's still going on right now, but we find that there's things like temperature dependence, there's like water dependence. Management practices can be positive, negative, or neutral impact on the species richness. We also have this time dependency. Then who has been here before? And these all might vary by all the different species that are coming in. So we start thinking about, like, what does that Y plus the management look like? And so we can write out a rough Stan model, and I actually hand this off to the stakeholders and say, why don't you write this, and we'll go back and forth on it. So this was like the first round where I basically sent over a skeleton code that's not here. And we sit around and talk, and then the stakeholders writing in the priors. So I said, like, this is, you know, probably like, this is just the start. This happened last week. So we eventually want to build up the model so we have what amounts to an ANOVA that's well informed, with all the priors in there and the priors recorded about where they came from. And then that can go to the manager, and then we can start thinking about things like time series models and occupancy models. But this was like that first step, an ongoing conversation, and that's what I'll be doing this afternoon, is getting on the phone to set up a meeting so we can go back over this and see what new priors have been set in. Are there things around like alpha and beta that need to go in there? Are there upper and lower bounds that we need to put in different places? So this also requires a leap of faith that somebody's going to follow through in that model. And you also have to put aside, oh my God, I can't believe they wrote that, right? So, or like, this is totally wrong. You have to set that aside and then just have an ongoing open conversation around model building. All right, next slide. So summary, you want to make this process stakeholder centered. You want to listen and translate things into priors, even if somebody's like, you're going out with somebody's abuelita, right? And she's saying, hey, these things were here at one point in time. That becomes one of your priors. So this, and again, the models of discourse, even when you're making bold statements, you get better and enthusiastic mutual understanding with writing Stan models. Like, you follow the workflow, and then you take the norms that are in the Stan discourse community, and you apply those to every single stakeholder interaction that you have. And I like leaving blank spaces in the models. You want those filled in definitely before you start running, right? Because you don't want your prior just being uniform. Is that when you lead to disaster? And I often talk about that with the stakeholder, like, this is why you don't want to do this, because we know stuff about the system. And you often hear in the Stan discourse, we have multiverse of models. You want all these multiverse of models, but they're actually a multiverse of different stories and different ways of telling this same story. And that's fascinating to me. You have this narrative that's building up over time with your stakeholders, and that leads to better buy-in. And it's a sort of a storytelling focused approach to research. And next slide. So the most important thing is to set aside your own priors. These are internal priors. This is why you see a land acknowledgement. It's why you see me saying I'm a queer New Mexican, because you may have certain ideas when you see the way I look right now. You need to set those aside. This picture is New Mexico. It's not that far from my house. It's about an hour and 20 minutes away. When you think of it, the environment and the communities that you're walking into, you got to set aside what you have in your head and be ready to have experiences like walking through this stream with some crazy volcanic rock. hanging overhead. So, well, thank you for your time. I know this was not terribly on the technical end of things, except for maybe the little occupancy model, but this is like how we interact with our stakeholders in the Middle Rio Grande.
Thanks so much, Era. We have some time for questions, and we have one that's come through the Q&A so far. Before we jump in on these questions, I'd just like to let the attendees know if some of them may not have come to a Zoom webinar before. So we've got the chat, and you should also be able to find a Q&A. And if you want to ask a question of whoever is speaking, sometimes it's most useful to type that in the Q&A rather than in the chat. That lets us keep track of which questions have been answered, which questions haven't, and so forth. So Era, the question from an anonymous attendee is, hi Era, building priors with stakeholders looks like a neat and important exercise to utilize local ecological knowledge and include diverse voices. Have you received skeptical feedback from reviewers about the priors being used? And if so, can you speak to this? Thanks.
Yes, we get a number of different reviewers. This often goes through different agencies like Army Corps of Engineers, Forest Service, USGS, that's the United States Geologic Survey. And then like from reviewers that are, like for journal reviewers. You do, because incorporating narratives as part of your prior, even though that's what we're doing with priors anyways, seems weird to people. It's like the fact that you would take somebody's spoken word and translate that into a probability distribution. But what we've done to address that is to show how rigorous it is and how we document, and basically the documentation of it. So like if we know that there's going to be a certain response to the variable that we're interested in studying, we can, if there's previous work that's in the journal, we can cite that. But oftentimes we're like, well, we polled experts, right? So we'll use academic language, which is not always the best thing to sort of give into, but it can often let you get your priors through the door if they're largely based on narratives, field notes, or just stories that people are telling you.
We've got a question from Dalton Hance. He says, great talk, Era. Have you ever considered building something like an interactive prior predictive check, for example, with Shiny?
Oh yeah, I've started to play around with that, but my Shiny R skills are not that great. It's on the list of things to get better at, because I see people doing that and it looks super cool. Just to not have that time to dedicate to that at the moment.
And I think we've got time for one more, and it's a good one from Ben Bolker. Can you speak a little about the details of translating the spoken word into a specific parameterized probability distribution?
That is challenging, because sometimes you have really long stories. You're out wandering through an environment all day long while trying to record data, take field notes, and listen at the same time. The best thing I found was actually using those index cards, because that captures, and unfortunately sort of renders the whole story into just a single distribution. When possible, we actually try to write down what's being told so we can be a little bit more, I guess, true to it, to capture that whole experience. I know it's not a very satisfying answer, but as soon as you clap things down to a distribution, which is what we need to do, you're losing a lot of the nuance too. Of what's actually being said. Ideally, you would be recording in the field all the time. We've done this on a couple of occasions, but some places where we go, recording is not allowed.
Wonderful. All right. Well, I just wanted to say, Era, you talk about how this is a less technical talk, almost apologetically, but this was wonderful. And I think we really need this as a community badly. So thanks so much. Just so people know, feel free to continue typing questions into the Q&A. We can continue to look at them and answer them in writing as we keep talking, and we can also save some for the panel discussion, which will follow the talks. And with that, I'm going to stop this share and turn it over to Harold Eister, who will be talking to us about some of his occupancy modeling adventures.
Thank you, Jacob, and great talk, Era, and I'm excited to follow you. Thanks so much for organizing this talk, Jacob. It was great to see everyone here, and I'm excited to talk to you about some things I've been working on. Just share my screen here. Can you all see that?
Yep.
So my name is Fari Leicester, and I'll be talking about using Stan to fit some high-dimensional multi-species abundance models. I'm currently a postdoc at UVM, which sits on the traditional ancestral territory of the Abenaki peoples. So as ecologists are often interested in knowing what species are where and why they're where they are and how many of them there are. And this has important implications for management and for trying to support biodiversity. And so some of the ways that people often do that is through occupancy models, which use data that's for in presence absence, or abundance models, which are useful for data when you have data on abundance. And some of the neat things about some of these models is that they can account for imperfect detection. So they can try to infer what the true abundance is, even if maybe when you were looking for the species in this area, you didn't actually see all of them. And you can also use these models to infer the abundance of species that might be rare or that you might have not seen very often by partially pooling data across a whole bunch of different species. And so I did some field work on birds in the US Midwest across 7 habitat types and 13 regional sites across the Midwest, and found 78 bird species. I'm interested in using some models to estimate the effects of these various habitats and localities across the Midwest on bird diversity and abundance. And as you can see by the number of habitats and the number of regions and number of species, these models will have a lot of parameters. And the way that people usually approach occupancy models or abundance models is using wind bugs or JAGs. This is what a lot of the published research resources are on. There's a lot of resources available to try to fit these sorts of models, including some really great books. And if you look through a lot of ecological literature, you'll see that people often are using something like JAGs, often through R. And so if you're doing some of this work, it's really attractive to try and use some of these well-established modeling techniques. There's a lot of resources out there, a lot of people who are using them, and a lot of kind of established knowledge about how to use them. But unfortunately, these JAGs and wind bugs have some disadvantages. For example, the samplers that they use can struggle to estimate models that are high dimensional, that have lots and lots of parameters. And because I just showed you these multi-species abundance models have lots of species across lots of different covariates, and so you can end up with a lot of different parameters. And so these Gibbs samplers, it's suggested, aren't as good at actually estimating well when you have such high dimensional models. And also because of the ways that these samplers work, you often need lots and lots and lots of iterations to actually get good estimates. This can take a long time to run. And these models often have limited diagnostics, which can make you think that maybe you have the right answer when actually there's some pathology in your model that you're not accounting for. So Stan can respond to lots of these things by Stan is faster and the algorithm is more accurate, so you can use fewer iterations, so it can be faster. Stan also, its algorithm is better at getting good estimates for these high dimensional models. But there's not very many people who actually use Stan right now for these sorts of modeling techniques. So there's not a lot of resources, books or otherwise, for fitting these sorts of occupancy abundance models. But luckily there have been people who have translated some canonical models in JAGs into models in Stan. So I'd like to thank Bob Carpenter, Rich Erickson, Hiroki Ito, and Jacob Socolar, who some of their translations were really useful as I was trying to figure out how to fit a model that's often fitted in JAG or wind bugs into one that's in Stan. And so because I was looking to create this multi-species abundance model, I couldn't actually find any example models in Stan. So I'll be talking a bit about this model that I created and perhaps how others can use it. And so the model, I won't go into too much detail, but you can see there's a lot of parameters. This kind of standard abundance model where you have two different models at the same time. You have both a abundance model that's the ecological process, and then you also have a detection model, which is modeling your detection of these different species. And in my case, these true abundance of these species vary across the habitats, those 7 different habitats, and across the clusters, these point count clusters across these Hubble states. And so, as suggested, I first simulated data that had all the features of my data. So it had these same covariates and similar sample sizes. Then I wrote a simple model in Stan, and then slowly adding more complexity, slowly adding covariates, adding in variation across different things, while also adding that same variation to my simulated data, until eventually I had a model that was could run well on this simulated data and produce the parameters I initially put in that data. And lots of people who are coming from other modeling techniques, we kind of expect that, and certainly when I initially came to Stan, you kind of expect that, oh, I'm going to take my data and I'm going to write this complex model, I'm going to fit it all at once. But for a lot of these Stan models, that doesn't work very well. And so it's a very iterative process where you're starting with a really simple model, and it often takes a while to actually get up to the model that you kind of imagined at the beginning. It's a very iterative process, and for me, I always there's lots of failures throughout that process where the model doesn't work, it's not giving the answers you want, it's not running, it's giving you all these errors, etc. But know that that's just really all part of the process. And so one of the advantages of Stan is it does give these really nice diagnostics. And so one of the key ones is divergent transitions, which is basically telling you that the sampling algorithm isn't able to find the samples that are better, and it's kind of failing to explore your probability space in a way that's effective. And a key way that you can often use to resolve those sorts of issues is by using a non-centered parameterization. So that's what I ended up using in my model, where you basically separate out your standard deviation and your mean from your actual normal distribution. And by doing that, you're able to help Stan estimate your model much better. And there's a lot of great examples on Stan resources available to try to find that non-centered parameterization for your parameters. And also, as one of the questioners commented about Era's talk, these prior predictive checks are really wonderful for making sure that your priors are doing what they think they are. I know for me, often these priors are on parameters that are in a different space and are about part of the model that really isn't observed, and can be a bit confusing to know if what you're doing is actually leading to the prior that you have in mind, or that your stakeholders have in mind. So using these prior predictive checks and simulate data can really help verify that these models of priors are the correct ones, and especially when you're using occupancy models, which are often in transform spaces. And so if you use kind of standard normal priors or other things for some of these transform parameters, you'll end up with really informative priors that are not informative the way you might imagine. And so this is really helpful to make sure that your priors are doing what you think. And so by doing this, by collecting this bird survey data, by building this Bayesian model, then you can get a whole bunch of iterations of your different parameters, and you can use those to simulate posterior communities, to estimating the true abundance in each of those locations where you counted birds. And then you can use that to get something like a diversity index, at your different habitats. So here I can use this data to show that these polyculture habitat farms where I was surveying had much higher diversity than these conventional corn, soy, and hay fields where I was also doing surveys. And so you can see the real value of some of these models. And this is the way that often Stan output or other output from Bayesian statistics is often used, where you estimate that posterior community, the true number at this particular spot in Wisconsin or Michigan or Illinois or whatever, where I was doing these bird counts. And then maybe you're interested in some additional analysis. You're interested in some analysis of the effective habitat, or you're looking at some functional trait, something. And so you might do additional analysis on those posterior communities and then get some credible interval at the end. But the problem with this sort of analysis is it actually ignores, for me, kind of one of the big benefits of Bayesian statistics and actually building up these structural models of your system. Because by building communities at those particular points where you're sampling, you're actually neglecting a lot of this model structure that you created. For example, instead of trying to simulate, this particular habitat, this particular location in Illinois, which then plates both this particular point across the region, this particular point in Illinois, not this particular point in Wisconsin, and then also the fact that it was a woods or a prairie. And so you're actually inflating this regional variation and this local variation across habitat types. And so then when you're doing initial analyses on top of that, you're not necessarily able to isolate the effect of regional landscape versus habitat type. And so instead, you can actually use your model to simulate communities that only vary with respect to one of these variables. So they can only vary with respect to habitat or only vary with respect to location across the Midwest. And so by building these by simulating communities that don't really exist, but that portray the sorts of your modeling structure, whether that's your habitat or the regional landscape in my example, then you can do initial analyses on those communities and come up with a downstream estimate of some interval that will reflect exactly the variation of interest. And so the takeaways from this are first that Stan can quickly fit multi-species abundance models and that the diagnostics can really help ensure model fit. And that I think Stan offers a lot of opportunities to kind of shift away from some of the JAGS and WinBugs models for multi-species abundance models and to start using Stan for those. I think Stan offers a lot of really helpful tools, particularly their diagnostic checks. And secondly, that once you actually create this model and you have this output, that this model structure that you have within your model can really help efficiently propagate uncertainty to downstream analyses at particular variations and particular levels. So the next step for me in using this model is to work on some data from along an elevational gradient in British Columbia. This past summer, Ben Freeman and I and some other people started this mountain bird network, which is seeking to use community scientists on eBird to collect data across elevational gradients. And so I'm looking to use now these multi-species abundance models to estimate the effect of elevation on these different bird species and to look at different species elevation niches across this mountain range, in the hope that in the future people can do additional bird counts and see how these elevational niches are changing. And so the problem I'm working on right now is to try to understand how to handle a different number of visits per site. So right now with the current data from the Midwest, I had the same number of visits to each site. And so you could use a simple array where you have species, visits, and sites, and you can feed that into your Stan model. But when you have a different number of visits per site, maybe some sites have 1 visit, some sites have 10 visits. Next problem I'm working on is actually how to try to incorporate that sort of data into some of these multi-species abundance models. And if anyone has any ideas for how to do this, I'd definitely be interested in that. Thanks to everyone for watching, and thanks to some of my collaborators who helped me with this project.
Harald, did you do all those illustrations yourself?
I did. That's try to combine my love of birds and illustration with some of my science. So it's nice to be able to have these opportunities to put it together.
Got a question coming through the chat. He just asks, when in your model definitions, is capital N a parameter and lowercase n data?
Let me just verify. That's correct.
Awesome talk, Harold. That was great. I'm going to leave it. Oh, good. Another question. So he says, how do you? Here we go. So how do you implement that without latent discrete parameters?
Yeah. So latent discrete parameters are one of the big struggles of doing these abundance models and Stan. In my case, I didn't have point count specific parameters, and so I basically was able to just estimate across all of my locations within all my point counts within a particular location, and so I was able to solve that problem that way. But when you have count specific parameters, then that sort of method doesn't work. And Jacob actually has, and Max Joseph also have some great write-ups about how to handle marginalizing these parameters in other contexts if you have count level parameters. And maybe Jacob will talk more about that. I'm not sure.
Hey, you have a question in the chat. So Daniel Hawking says, since abundance becomes a derived parameter, how do you estimate n so that it is always greater than or equal to count?
Yeah, so you actually, when you're estimating, I guess I'm not sure I understand the question. Always greater than or equal to count. So you're actually, when you're when you're actually building a model, you're actually first taking the maximum number of individuals that was observed at a particular location, and then you're actually when you're doing your modeling, you're actually starting with that max number and going up to some number k, which you think is the maximum number that could be at all of your sites, and so that way you're ensuring that you are getting the max, that n is always greater than count. And some of Bob Carpenter's models, that are on these, that are some of the example models, show how that's done.
All right, awesome. I'm going to start getting set up for myself, but everybody feel free to keep peppering Harald with whatever questions you have.
I guess I have a question for you. So there's the community of people using JAGs and bugs, etc., but there's also Nimble. Is there ever any hesitation to say, like, you doing something specifically in Stan rather than other software that people might use?
Yeah, I guess for me, Stan is a bit more flexible for lots of the other problems I'm working on. And so it makes sense to kind of invest more time into learning that and to use it for all the different problems I'm working on, some of which those documents models, some of which other ones. So for me, it made sense for that, and not because just see the flexibility. But certainly I'm sure there's lots of other particular problems where other software is better if you're kind of specialized.
Can I just double check? Do people see my screen now? A PowerPoint occupancy model for Columbia's bird life?
Yep.
Awesome. Well, then I'm gonna I'm going to get cracking. I'll leave my video off. I'm coming at you from my bedroom in Ithaca, New York, where I'm a postdoc at the Cornell Lab of Ornithology. The work that I'll be talking about today, though, was done during my previous postdoc at the Norwegian University of Life Sciences. And this is going to be a story of my life, really, which is trying to push Stan, well, not push Stan, but push the frontiers of how we collect and model data. And I work primarily on birds, but on birds and in ecology. And that sort of was a very natural then for me to land with Stan. And this relates as well to the previous question about Nimble. The promise of Stan, sort of as Michael Betancourt presented it years ago, is that we can use this HMC sampling to push our models into a space where we have really complex models informed by really big data. And I think Stan is sort of uniquely good, or HMC, I should say, is uniquely good at doing that. And for those who might not know or be as familiar with Stan, HMC here is the particular Monte Carlo routine that Stan uses under the hood to fit models. This work was done with a lot of help from a lot of people, especially with the fieldwork component, which I won't mostly be talking about today, but Simon Mills up here at the top not only did a lot in the field, but also was really instrumental in helping with some of the modeling that I'll be talking about. And then I'll just put my affiliation and contact details up here as well. I guess most of you know who I am. So the roadmap for this talk, I'm going to talk to you just a little bit about the study system in Colombia. I'm going to talk to you about what I call the biogeographic multi-species occupancy model. I'm going to tell you a little bit about how I fit it, and then time permitting, a little bit about a new R package that I think might be pretty interesting to some of you. I am cramming way too much into the time available, so we'll see how far I get. I'd just like to start by telling you a little bit, though, about what occupancy models are, what we're talking about here. And I don't have time to go into great detail, but I want to mention explicitly, you can see down here at the bottom some links that I have. I've written up quite a bit about what this model is and how it works and how to implement it in Stan at the links below. The first one is sort of a more basic introduction to the model. The second one covers some more advanced topics related to how we think about the likelihood. And the last one is the R package that I hope to have time to tell you about. But the basic idea behind these models are that we partition observations of species at sites into, we partition the probability of observing a species at a site into one component that's related to the probability that the species is there, and into a second component that's related to the probability that we will encounter the species on our visit, given that the species is there. And it turns out that by constructing our sampling design to include repeated visits to a single site, or to many sites, I should say, we can identify, that is, distinguish between these two parameters and model them separately, which is really useful because the factors that govern detection are not always the same and can be confounded with the factors that govern occupancy. And increasingly, those of us who study whole communities like to do these models in a community context, which is to say we incorporate random effects of species in the model itself so that we can pool information about occupancy and detection processes among species that are assumed to be similar, or if not similar, species that are assumed to be exchangeable, conditional on covariates. So we can incorporate species traits, for example, or phylogeny into these models. So to shift gears a little bit and talk about Colombia. Colombia is one of the very most biodiverse countries on Earth. And in particular for birds, it is the most biodiverse country in terms of avian richness on Earth. It's got well over 1,900 species of birds. And even if we exclude those that are marine or strictly coastal, we still have upwards of 1,800 species of birds. And the biogeography is really dominated by mountains. So this is a map of elevation. Paler colors are higher. And this is the main Andean chain sort of swooping up from southwest to northeast. And it splits into 3 subchains that are separated by 2 really deep valleys. And then up here in the far north, we have the isolated Santa Marta Mountains, which are the most important center of continental, non-island center of avian endemism in the world. And across this landscape, we've got all kinds of species with all kinds of ranges. We have species that are only in the lowlands on one side of the Andes. We have species that are only in one of the valleys. We have species that are everywhere in the lowlands. We have species that are everywhere in the highlands. We have species that are only in a tiny piece of the highlands, or species that are only on one of the highland ranges. So the biogeography is really complex. And of course, I'm showing you 6 range maps, but actually there are 1,800 of these maps. And because this is pretty dry, I just want to remind you that these are amazing birds. So this map here is for the sooty ant tanager, which is an endemic of the lowlands, the humid lowlands of the Magdalena Valley. Within these landscapes, the predominant land use change across most of Colombia, or certainly a ubiquitous land use change across most of Colombia, is cattle pasture. It goes from the bottom of the mountain through the middle of the mountain to the top of the mountain. And we were interested in really trying to understand at scale how bird communities across Colombia are responding to cattle pastures. And so what do? We go and do a very, very large amount of survey work, which I'll talk in a bit more detail later. So we have information about bird communities sampled in forest and pasture at 848 points. Most of these dots represent multiple points across Colombia. And then we also have, well, let me back up. We have a real challenge, though, if we want to take data like these and model the species in a multi-species context. And that's because the species ranges are so diverse and different that it's a big challenge for us to implement a covariate structure that's sufficient to distinguish all of these different forms of range. If we fail to constrain species ranges properly in this model, then first of all, we'll get bad inference. We'll say that species are in places where they aren't. And second of all, we'll get bad pooling because we'll be pooling intercepts that reflect a species like this tropical kingbird down in the lower right. Its occupancy intercept would be relatively high because it occurs across most of Colombia. Whereas this Cundinamarca antpitta in the upper left would get estimated with just like a sort of stupidly low occupancy intercept because it's found in virtually none of Colombia. And without a covariate to distinguish what's in range from what's out of range, we get these sort of smeared out uniformly low probabilities for range restricted species in particular, which turn out to be of real downstream interest for us, although I won't go too deeply into that or why that's the case. And so the solution for us to get good principled pooling across these species with disparate ranges is to leverage this a priori range information that we have for the species. We call this a biogeographic multi-species occupancy model. And I'm going to show an example in the context actually not of Colombia, but just an example from the North American Breeding Bird Survey. So here's an occupancy model. It's actually the model of Jarzina and Yetz 2018, for those who care, that's fit to the warblers of North America. We've got prothonotary warbler on the right and red-faced, on the left, and red-faced warbler on the right. And the model has basically a term for elevation and very little else. And so prothonotary warbler is commoner at low elevations. And so the model estimates that it sort of could be anywhere at low elevations. Here it is in the east. Here it is in the Central Valley of California. The red-faced warbler tends to occur at higher elevations, and the model picks that out. It's showing up with some occupancy probability in the high Rockies, although it's difficult to see. But these are both badly wrong maps because the ranges of these birds are not reflected. So this is what you get if you don't have a sufficient set of covariates to constrain the range. The challenge is that these, so here are the ranges from bird life on the bottom, the breeding ranges of these birds. And the challenge is that there's no way that we're going to come up with a sufficiently rich set of covariates to properly constrain the ranges of each of these birds, including those that have limited data in the first place. For example, this red-faced warbler, which is really just restricted to the higher mountains of the far southwest. What we can do instead is we use these range maps to create what we call a range covariate, a covariate that reflects the extent to which a bird is in range or out of range by a particular transformation of the distance from the range margin, which I won't go into in great detail other than to say that when we include this range covariate, all of a sudden we get really beautiful inference. This is a pretty darn good map of occupancy probabilities for prothonotary and red-faced warblers in the US. Not only that, but we can also then reject a lot of sort of the zeros, the lack of occupancy probability far from the range as sort of uninformative structural zeros. We can clip all of those points out away from model fitting, and we still get good inference for the in-range points. So this is what we call the clipped biogeographic multi-species occupancy model. Sometimes the clipping can even improve the fitting for the in-range points. So I'll show you one more example from the breeding bird survey. This is mourning warbler. It's a northeastern species that occurs at higher elevations. Here's the naive model. Here's what we get once we have the range covariate. Here's what we get once we do the clipping, and it's a little hard to see, but what you can hopefully spot is this patch of high occupancy probability in West Virginia that's getting modeled in the clipped model, but not in the unclipped model. And the reason we see that, well, here's what happens if you take this clipped model and project it across all of the United States. What's going on is that the locally appropriate elevation relationship for the Northeast is so steep that it overcomes the negative distance relationship and yields high occupancy probabilities across the Mountain West. So B differs from D because it's had to estimate a much shallower elevation relationship in order to avoid this blob of erroneous occupancy probability in the high mountains. C, the clipped model, doesn't have that constraint. So we're going to now take this model and apply it to Colombia. I need to tell you a little bit about the data in Colombia that we're going to be using. We've got so 848 points across Colombia, and you can notice again, most portions of Colombia's biogeography don't have nearly enough points in them for us to actually constrain an a priori model of a species range. You know, we have Cundinamarca antpitta on like 2 points. Is that because it's range restricted, or is it because it's just rare? Well, it's because it's range restricted and it's actually common in its range, but our data by themselves are not sufficient to tell us that. Another key feature of our data that you need to know about is that they're organized in clusters of 3 sampling points that each sample either forest or pasture. So this little circle here reflects these 4 clusters, 2 forest clusters and 2 pasture clusters that are matched for elevation. And then we have this wonderful range data, which comes from Fernando Ayer de Quiñones, who wrote a field guide, but also distributed the range maps that underlie his field guide, which are incredibly good, like really high quality range maps, better quality than exists for most places in the world. And he also published elevational max-min ranges, so an approximate elevational bound for each species in Colombia. And so what we do with these elevational ranges is we take a species minimum and we take its maximum and we transform the elevations of all of our sampling points for that species so that the minimum corresponds to -1 and the maximum corresponds to 1 by a linear rescaling. And then we actually do an additional bit of biogeographic clipping just to speed up the model fitting at -3 and 3. So for this species, that would be at 500 meters and 3,500 meters. We say, look, this species just doesn't occur at elevations beyond these. And what this elevational range covariate then lets us do is, again, put all the species on a common scale, a common elevational scale, which among other things, enables principled pooling of the intercept. So now the intercepts in our model are going to reflect the probability of occupancy for a species in the core of its elevational range, in the core of its geographic range. And that's something that we can pool across species, whether they have narrow elevational ranges at the bottom of the mountain, or wide elevational ranges at the top of the mountain, or restricted geographic ranges or wide ones. We also do some biogeographic clipping, not just at some suitable buffer beyond species ranges, but also at the major biogeographic barriers of Colombia. So we don't allow species to cross the major mountain ranges or valleys when their range maps show that they're restricted by these barriers. So we've got 848 points, each visited 4 times. We have 969 species detected, plus 645 never observed species that are plausibly occurring in the vicinity of our points based on the biogeographic clipping. So another advantage of this biogeographic model is that it lets us fix the identities and the traits of all of the never observed species in the entire community. And so we get taxonomic information, and we're using 17 species traits in this model. And we fit this humongous multi-species occupancy model to the data. Occupancy is predicted by land use, distance to range, linear and quadratic terms of standardized elevation, 17 species traits, and the interactions of pasture with the 17 traits. We have random intercepts for species, family, species by cluster of 3 points. That species by cluster of 3 points random intercept is a parameter vector with 200,000 elements. So this is a huge, huge model. And without Stan and HMC, we just can't fit it. And then we have a species by subregion effect as well to deal with some additional spatial autocorrelation in the model. We have random slopes for species and sometimes also family, and also an elaborate detection submodel. We have some little tricks that we've used to make this work, and I'm actually going to skip a couple of them for the purposes of time. But one that I want to talk about a little bit is how we figure out priors. As Harold alluded to, we have these priors, vague priors on logit scale parameters induce really peaked priors on the probability scale, which is undesirable in these models. And in particular, we weren't so concerned about a big prior peak near 0 because there are a lot of point species combinations. For example, forest species on pasture points where the probability really is 0 or close to it. But this peak near 1 is really undesirable. That there should very rarely be guarantees of occupancy for a particular species point combination anywhere. So we end up using a non-zero centered prior on the intercept. The other thing that happens is that because this logit transform is so nonlinear, when we have random effects here, it ends up, so for example, if we have 25% of our points occupied, then we could achieve that with an intercept of, whatever it is, -0.5 or something like that on the logit scale. Or we can achieve it with an intercept that's much, much lower, but then a high variance random effect, because the tails of this random effect start to overlap the high probability region of the logit. And so we had some real challenges because we didn't have strong prior expectations about the random effect variances, particularly for these spatial terms, the species by cluster and species by subregion terms. And they ended up, what we ended up doing is just doing lots of prior push forward checks to the probability scale. And we ended up with a very low prior, a very wide standard deviation on the spatial effects, and then a very, very low prior on the intercept. Normal minus 7.5 mean and 2.5 standard deviation. And amazingly enough, we did all this prior predictive checking as carefully as we could. And when we fit the thing, we had no prior data conflict. We sort of nailed everything. So with that, I'm basically out of time, unless I've miscalculated here. But I do just want to quickly, we have a bit of a buffer built into the schedule, and I'm going to use it to tell you very briefly about this R package that we've built, which we call Flocker. We wanted students to be able to start using this model. And so what we've built is an R package that works as a front end to BRMS that lets you fit occupancy models using full BRMS formula syntax. I'm happy to talk more about it later. It can do anything BRMS can do with the linear predictors: splines, smooths, fancy random effect structures, phylogenetic models, spatially autocorrelated models, monotonic effects, you name it. And we can also then automate posterior prediction, posterior distributions for the unobserved occupancy state, LU-CV approaches to model comparison, and so forth. So check out our webpage for that. And I don't have time for questions, but I'll be happy to take any after Max's talk. I apologize for running a little bit long. You there, Max?
So Jacob, you have a question in the chat by Mark.
Cool. Yeah. So yeah, what we do with these elevational range covariates is we fit linear and quadratic terms. So we allow the quadratic term is almost invariably negative, and we allow or it is in fact invariably negative in this model. And the linear term, though, is important as well. We don't just square the range, we don't just square the covariate right off the bat, because some species really are more abundant in the upper part of their range. Other species are more abundant in the lower part of their range. So we want to have the linear term in there as well. And has anyone heard from Max Joseph that he's here and ready to jump in?
There you go.
All right, I'm here. Is anyone else here? Sorry, I got booted off there inopportunely.
No worries. Good to have you back. Take it away. Do you need a couple minutes? Which would be fine if you do. Can you hear me, Max? Ruh-roh.
Hello.
Can you hear me, Max?
I can hear you a little bit, yeah.
Okay.
Can you hear me?
I hear you right now, and I've been seeing you this whole time. You're ready to roll if you want to.
Okay.
If you need a couple minutes, that's fine.
Well, let me just share my screen here. Oh, I think you need to make me a co-host.
Oh, you got booted off and came back on. I see. That's the problem. I have to find you again and rejigger you to co-host dumb. Okay, you should be good to go for real now. You might need to unmute Max, or there might be some audio issue. Or maybe it's just me.
No, I can't hear him as well.
I'm not sure if he's hearing us. Let's
I don't think so. Hey, Max.
while we're waiting, still can't hear you. Well, I'll let you know as soon as I hear something. Or I wonder if it could be the headset.
We could hear you before you started sharing your screen, if that helps with anything. No. No. Yeah, so nothing yet.
Sometimes if I manually adjust the microphone input on the audio settings, sometimes that helps.
I've turned on captioning, which I should have done from the beginning for accessibility. I apologize for not doing that. I wonder if you could try saying something, Max, and we'll see if we can see it typed out. No. While we're waiting, I'll answer two questions that came over the Q&A. Andrew McDonald asks about making predictions for range shifts using these biogeographic multi-species occupancy models, as well as whether there's a circularity between using the ranges to inform occupancy well, let's see, can we hear Max? Oh, maybe I have to make him a co-host, make co-host. And then.
Can you hear me now?
Heck yeah.
Oh, beautiful.
Let's see what happens when you share though.
Yeah. Okay, perfect. Thank you all for bearing with us as we sort out these audio issues. I will share my screen and jump right into it.
Still working.
Okay. Beautiful. All right, so today I'm going to talk about MLOps in a Bayesian workflow. And specifically, I'm going to focus on how you can track experiments, which it sounds like we all do, using a tool called MLflow. I'm currently a data scientist at a company called NCX, and there we run a carbon offset market. And one of our flagship products is Basement, which is a high resolution, tens of meters Bayesian map of forest carbon in the continental US. And so when I say Bayesian map, what I mean is for every pixel, we have a bunch of draws of forest carbon. And building this model, as you might imagine, is not super easy. There are a lot of challenges of scaling that we run into, and experimentation is something that we do to find models that are efficient, that we can actually deploy and transact upon. So quick outline here. My perspective is, coming from the academy, recently jumped into industry. And so MLOps is something that's like new to me, but something that I think pairs really well with a Bayesian workflow. So I'm going to give a quick overview of those two things, focus on how to track experiments, and then get into a quick demo. So somebody mentioned this Bayesian workflow paper earlier. It's worth a read. This is essentially a decision tree / conceptual framework that you can use to build trustworthy models. And so you sort of start by picking an initial model, checking that your priors work, checking that the computation is valid, checking that the model works well in a few different senses. And the path from initial model to model that you trust that you want to use is usually, iterative, right? We go through lots of cycles of tweaking the model, in some cases, tweaking priors. You may get different data. And so there are a lot of experiments in the process of going through this decision tree. And tracking those experiments is pretty hard to do. Now, MLOps is a framework that's a bit broader, and it really relates to how do you build, deploy, and monitor models in production. So a lot of us are probably most familiar with this data analysis, model analysis, data science experiments part of MLOps. But once you decide on a model that you want to use, to actually deploy that and make predictions on the fly and monitor performance, those are some of the data engineering, model engineering tasks that may be less familiar to people. But what I'm going to focus on today is this sort of data science experiment side of things. I think that's where many of us live, and certainly where the Bayesian workflow really comes into play. So let's think about injecting a Bayesian workflow into this part of MLOps. How do we actually keep track of all of these experiments that we're running on the way to arriving at a model that we like? Over the years, I've seen a variety of strategies for keeping track of experiments. Some really diligent people that are more organized than myself may have notebooks that they write down, what experiments they're running and what they found, et cetera. You may use different subdirectories within a project directory to track different experiments, or maybe you're working on different Git branches or relying on the Git history to represent the experimentation that you did. Or if you're like me and you're just really unorganized, maybe you don't track experiments, you just do it and then end up with a model that you're going to use at the end of the day, right? And we all know that's bad, but I suspect it's not super uncommon. So here's what we came up with at NCX. And this is not my idea. This is really, there are some really great data engineers that we work with who helped us think about this framework. The way we think about it currently is we think about an experiment as one model run. Okay, so we start off our run by logging metadata, what is the experiment that we're running? And this might be, you could think of metadata what is the set of priors that you're using, or what is the likelihood that you're using. If you wanted to compare like a Poisson to a negative binomial, those are things about the experiment that you know ahead of time that you could log, for instance, logging the Stan file as metadata. Then you actually do sampling. We use Stan, but you could sort of use whatever you want in this step. Once your posterior sampling is done, you compute and log a variety of metrics. So this may be like expected log predictive density, the runtime, Pareto K diagnostics, leave one out diagnostics, R hat values, et cetera. You can think of these as like real valued metrics that provide some information about how the experiment went. And then finally, you can log artifacts, basically files that were generated in the experiment that you may want to revisit later on. So that might be the actual model object that you've created. It could be plots. If you're doing predictive checking, maybe you want to save out those visualizations. The way we do it is we actually save an RMD report that gives us a bunch of diagnostics all in one place that we can quickly scan and see how is this model looking. So that's like an experiment, right? And we want to track all of these things, and we end up running a lot of these experiments, and we would like to retain the history of that experimentation. So I am going to point you all to this GitHub repository. I'll give you a sec. If somebody wants to like put that in the chat, that would be awesome. And if you go to that GitHub repository, I'll show you it in a second. I just need to, my computer crashed right before hopping on this call, so I need to spin up my Docker container again. But there is a Docker file in there that you can use to walk through this later if you want. So I have that right here. And I'm just going to walk you through a quick example of how to use MLflow with Command Scanner. There are BRMS and just like LM examples in this repository as well. And let me zoom in so you can see this better. I'm going to work with the Penguins data set, which hopefully you all know. Essentially what we're going to try to do in this simple example is predict body mass as a function of bill length. So here's a scatter plot of what the data look like, colored by species of penguin. I'm going to predict body mass as a function of bill length. So this is my formula. I'm making a design matrix and then putting this all into a list that we can pass to Stan. So that's what that list looks like. There's the number of observations, the number of columns in the design matrix itself, and the response variable. So we're going to start a run, which is essentially like, hey, I'm going to start an experiment. Setting a tag is a way for us to keep track of like what the experiment name is. And then we'll just fit this model real quick. And while it's running, I'll show you what the Stan model looks like. It is just a linear regression, right? So we've got a vector of coefficients beta, standard deviation, sigma, some priors. This is our likelihood. And then I'm saving out this log-like object so that we can look at leave one out diagnostics and a few other things. Okay, so that's done sampling. Next, I want to log the parameters of the run, and an MLflow parameter is a bit different than what we usually think of in a Bayesian context. It's essentially like the metadata of the experiment. And then we can compute leave one out metrics using Command Stan, and then log those metrics using MLflow, right? So I'm logging the expected log predictive density and its standard error. Another thing that you might want to do is compute our hat values and save those out so that you can see, did the model actually converge? So I'm saving out the R hat value of this LP dunder object. And then also maybe you want to save out the maximum R hat, like what's the worst R hat that you got? I mentioned artifacts. So perhaps you want to look at trace plots later on. You can save those as local files and then log those as artifacts. And then you can save the model object. I won't get into this too much, but when you're done with your experiment, you end the run. And then if you look at the user interface for ML flow, it looks something like this. So this has your experiment. You can click on it, see the metrics that you've logged, look at the artifacts that you have. And if you want, you can reload this model object to interact with it later. And so one thing that is really handy about this framework is it gives you a really easy way to actually like run new experiments and compare your results. So let's say I just want to change the formula. So I've added an interaction term between bill length and species. All right, so this will give a main effective species and an interaction. I fit another model, and I'm just logging out the exact same metrics that I logged the first time. I can then look at the user interface once this is done. And that gives us a really easy way to actually compare the results from the experiments, right? And I promise this is the end of the demo. I'm just going to show one quick thing and then wrap it up. All right, so now we have 2 experiments. If you want, you can like compare runs really easily. So here are my 2 formulas. I can look at expected log predictive density metrics, R hats, etc. There's some built-in visualization stuff that is semi-useful. But the point is that all of this provides a really easy sort of principled way to do all of this logging without thinking about it. You just have to think about it when you're setting it up, and then later on it's baked in. And for people like me who like, I'm not diligent enough to write all this down in a notebook, I've found this really helpful. So that's all I've got. If you want to learn more, feel free to reach out. This is my email address, or you can catch me on the Stan forums at mbjoseph. Thank you.
Max, how heavy is the overhead, the first part of the learning curve, would you say, for sort of getting up and running and you know, getting the appropriate directory structure on your computer sorted out and all of that stuff?
Yeah, so directory structure doesn't really matter. It's kind of agnostic to that. The hard part for me was initially just figuring out like how to get ML flow to work. The installation is a bit of a pain because it requires Python. It's like linked to a Python ML flow, which is one reason that I built this Docker image. So like if you have a clean Python environment on your computer and you're using Conda and you're like, you know, on Santa's nice list, it might work out fine. If your Python situation is complicated, you may find that the installation is the hardest part. Once you've got it installed, the next crux, which is something that we probably already think about, is like, what do you want to log, right? What are the things that you would like to compare? What are the things that you'd like to track in your experimentation? And that's the fun part, but it's not obvious always, like, what are the things that you're wanting to optimize for? So for us, we're building really big models, so we care not only about performance, but also efficiency, right? So like MCMC efficiency is one thing that we track along with a variety of other cross-validation metrics. Oh, and I see Mitzi's mentioning there is a Conda installer for Command Stan Pi for Pythonistas and Command Stan as well, which is great. I see Max is asking, does it save all draws for each saved metric? Okay, so that's a really great question. We save all of the draws in the model object itself, essentially. So the metrics that are logged, the way that is intended to be used is like real valued metrics. So while you could log 2,000 draws for your beta coefficient vector, that would be a ton of metrics, and it would kind of pollute the MLOps browser interface and would be hard to work with. So what we do is we save the draws for sure as serialized model objects so that we can load those later. And it's actually really like, you have to have that to deploy a model, right? Because you want to make however many predictions as you have draws downstream once you've settled on your model structure.
Let's jump in and give people a little opportunity for a break here, I guess. Well, we can come right back to this. And there's no, so it will again become clear that I don't really know what I'm doing when it comes to organizing a Zoom meeting. But there's no real structure from here on out, other than that we want to have a panel discussion. I was not supposed to moderate originally, but I think I'll sort of be in a pseudo moderator role. My goal will just be to completely minimize the amount that you have to look at or hear from me from here on out. But if we need to move things along, I'll try to help do that. So let's reconvene in 5 minutes, but if you're not back in 5 minutes, don't sweat it, and we can get going and see where this takes us. We'll try to panel discuss for about a half hour, and then what we'd like to do, SBC suggested this, and I think it's really smart, is after the panel discussion, we'll just try to have some kind of a more social moment where the attendees can talk and mix and we can talk. Unfortunately, it turns out you can't do that over a webinar in Zoom. So what I'm going to do is fire up my own personal Zoom room and send everybody the link to that, so that anybody who wants to at about 2:15 can hop on over, and I'll have breakout rooms set up that we can just sort of see how that goes. And if it's terrible, we'll do something different next week. All right, I'll see everybody in 5. Well, I'm back. If anybody else is back, feel free to hop on. And I'll at some point introduce VNA as well, since that hasn't happened yet.
Yeah, I'm just crashing the party today, to be honest.
Well, I do really appreciate you being here, and also on fairly short notice for taking a more than attendee role. So everybody give a round. Well, let me go ahead and introduce you, I guess. In addition to our speakers today, we have quite a few experts in the audience, but one in particular who will be joining us for our panel, and this is Vianney Leos Barajas, who's on the faculty at the University of Toronto now. And maybe I'll let you just say like 2 words about who you are, and then figure out what to do next.
Yes, I'm an assistant professor at the University of Toronto between Statistical Sciences School of the Environment. I am Mexican, Mexican-American, grew up in California. Now I'm here. I am not an ecologist. So I always love actually coming to these talks. I get to learn so much from everyone here. It's so amazing. I also have a few cats, so love the cats. And also love everyone that's actually here in the attendees. It's so amazing to see such a wonderful community. So thank you. And thank you for letting me crash today.
And I'll just say to everybody, part of why I wanted VNA on this panel is particularly, you know, most of us are, or many of us are ecologists in some degree or other, but VNA really knows the stats. And so in as much as the discussions veer in that direction, I'm really looking forward to hear what she has to say. All right, well, we don't have any open questions right now. So I guess maybe the first thing we can do is just run down the panel and maybe everyone could just like pick a talk or a point or something they thought was interesting or anything they want to elaborate on. You know, just something that stood out, and we'll see if we can get a conversation sort of spun up off of this. As I said, I don't know what I'm doing, so I certainly hope that you guys are dynamic panelists, because otherwise it's going to be a drag.
So I'm gonna ask the other panelists a question. I also love the cat. So from a statistical standpoint, I kind of, I love HMC, I love all the technical details, etc. But I've often wondered, how ecologically realistic should our statistical models be? Right. So you have all these amazing dynamics in your ecological data, and we're talking about this like posterior predictive checks to assess the model capacity. And I've noticed a lot of statistical models can, they have great residuals, your PP checks all look good. But once you start sampling from the model, you have such complex dynamics in your ecological systems that you can't really capture everything. So to what extent do these statistical models need to reflect our ecological realities?
Max, why don't you take a crack at it?
Yeah, sure. I'll take a stab at it. I think that's a really good question. From my perspective, the quantitative ecology community has done really well in the past decade in terms of building realism into our observation models or our likelihoods, like thinking deeply about how the data are collected and reflecting the sampling design and protocol in like a somewhat fancy likelihood function. There's been less focus on like the actual ecology part, right? Usually it's like X times beta gives you expected abundance. But there is this other domain of like, you know, really deep model-based ecology that is often less tied to messy, large-scale data sets. In some cases, like maybe purely theoretical or microcosm, mesocosm based. I think bridging that gap is hard. In some settings, it's like easier to know the answer in terms of how much ecological realism is necessary. Like for instance, in my current role, what we really care about is nailing the estimates of forest carbon, right? So if our models can do that, it doesn't matter as much like how well they're capturing competition dynamics between individual plants, right? Like we kind of know the targets for precision that we need around carbon, and we care less about reflecting ecological reality there. But I think there are other settings where those details actually would matter a lot. Like if you're thinking about managing one species that is preyed upon by another species, you kind of need to know something about those interactions to understand the consequences of decisions that you would make later on. But it's not easy.
I think for us, we have those like species interaction things where somebody wants to go do some management action and it's good for this species, but it might devastate this other native species and allow tumbleweed and other things to come in. So we have to get those, but then also depends on the scale of the management. A lot of times our data is collected on these sort of like small scales and these like 100 meter by 200 meter plots, and they want to do something in what they call like a reach level, which spans tens of kilometers sometimes. So a lot of what we do for our models is sort of driven by that. So like how realistic is it when we basically are doing like post stratification on our ecological models, try to figure out what's happening at the, the tens of kilometer reach. What's our tolerance of uncertainty? And that's often dictated to us by the different funding agencies. So sometimes we can disclaim responsibility, but at the same time, if you're thinking about wanting to publish this, you're like, well, yeah, how realistic is this? And what can we do to, I guess, roll more process-based models into something that's largely just a correlation model?
With apologies, I can't resist jumping in on this particular question as well, just to say that as we push the data big, the posterior predictive checks get bad. Everyone who claims that they have good posterior predictive checks is either not implementing a sensitive check in ecology, is either not implementing a sufficiently sensitive check or just doesn't have enough data to detect the misspecification. And of course, there are times when we need a simple model, when we don't have enough data to support a realistically complex model. But when the data support it, I think that's what Stan is great for. By all means, try to wrap as much process and as many subtleties as the data are able to support, because now we have the computational tools to support doing that. A question coming up from the chat. For anyone who's created hierarchical ecological models with an observation model in addition to a process model, just how do you validate the partitioning between the process versus the observation error? I've been thinking about a comment from a colleague made recently when they used simple mixed effects models in the past, where a lot of variation was soaked up by observation error. And now with new models, a lot of it gets chalked up to process error, but the data are the same. And I might kick that one to VNA if you feel able to give it a crack.
You're able to tell very quickly that I'm also not a very good statistician. I think with the errors, I think it's another thing with these statistical models, like how much, what can we actually capture? So we can always write out the math. Right? It's easy to write out the math and say this is this beautiful model that we want to fit. Once we generate data from that model, you actually might not even be able to recover the error terms or disassociate the error terms. So I think it's one thing that's great about Stan and the Bayesian community is that it reinforces that there are these models and we can generate data from them, from known fixed parameters, and realize quickly that we might actually not even be able to estimate that same model with the true data. And so in this case, if you're trying to decompose the error into observation error, process error, it's the limits of identifiability or imposing some type of structure through prior specification saying, I know this. And in order to actually capture the errors where they should be, where I want them to be, I might have to impose some limitations, some orderings, something of the sort. So I think prior distributions and really understanding the mathematical object and the limitations of how well you can recover it with how much data. That's usually what I suggest to people, mostly because the models that I fit, I also have a lot of problems with. So hope that's helpful, but I'm sure others here.
As Jacob is reading that question, I can see all of us were smiling because I think we've all experienced that problem. And I think I definitely approached it the same way that Danny was talking about. We're kind of using prior specification to put in maybe a bit tighter bounds on what is reasonable to make sure that even if maybe the data isn't enough to, on its own, figure out what error is in which spot. Often our priors are, in my case, my priors were, even with just like kind of reasonable priors, it was enough to kind of shift the error into something that made sense. Because I had the same kind of thing where if I simulated data with the same sample sizes that I had, you weren't really able to recreate those parameters, but using priors you were.
Yeah, and I see everyone else sort of nodding their head, so feel free to chime in and just, we can all talk about our failures together.
Yeah, I guess so. This is definitely something that I have struggled with. For instance, in like an occupancy modeling context, if you want to know how well your model captures features of the data, that's pretty easy, right? You can, well, not easy, but you can use predictive checks to see how things stack up to the actual observed data. Validating the latent part, like the actual unobserved states, is harder, right? I mean, there are some clever tricks, like I'm thinking of in a distance sampling context, the latent thing you care about is like how many birds are within some radius of this point. And if you just walk out to a field, like you probably wouldn't know that with certainty, but there are experiments that people have run where they like plant speakers and they know how many speakers there are in this radius, and they can basically like remove all uncertainty from those latent components. And in doing so, evaluate like how well the model can actually capture or reflect the latent data in the same way that you would normally do with observed data. There aren't always obvious tricks like that. Like if you're trying to count fish in an ocean, it might be harder to scale something like that. But yeah, that is something that's really hard. Mevin Houghton once described this like as a balloon animal type of concept, where if you only have an observation model, like a mixed model, and you're ignoring this hierarchy between the observation model and the process model, a lot of the variance will be associated with the observations. But if you then build in that hierarchy, you can think of squeezing the balloon animal so that some of it goes into the process component and some into the observation component. Which one is right, I think, depends on a lot of it. It's like your modeling philosophy, like how much do you care about capturing the details of the process versus the details of the the data collection protocol, right? But yeah, that's another tough one. That's a great question.
I'm just going to read a comment that came through the
I
chat.
handle that
From,
stuff in,
oh, sorry, go ahead.
so I handle pretty much the same way as everybody else, right? Yeah, just lean into those priors. And sometimes like I'm not sure why I'm picking a certain prior to make that happen, right? Just like it seems to do okay, but a lot of times we don't have good constraints in our system either. It's just a high amount of uncertainty. It's like we know there's a relationship between depth of groundwater and plant productivity and animal species in the riparian habitat, but there's a big sort of latent variable that like what's actually connecting that, and we want to sort of get at that, but we don't have an easy way to actually sort that out. And so then we lean in the priors and maybe just go, as we start bringing in mathematicians and collaborators to help us try to narrow down some of the priors to something reasonable, as opposed to, kind of guessing like, oh, this looks nice and it, makes the model perform well, but is that the same thing as us getting at what actually is happening and how it's partitioned?
I want to read quickly a comment from the chat from Dan Avando, who I want to read it just because I think it's actually insightfully touches both on this question and on the previous question about sort of levels of complexity and realism in the models. It was written in response to that first question. And he says, I think the answer depends a lot on the objective of the model. A model with a good causal identification strategy, but very low predictive power, can still be very useful if the goal is understanding the parameter. If the goal is to be able to predict the dynamics of the system, then the realism probably matters a lot more. And then, well, I'll just read the whole thing. I think it's good. How good does it have to be depends both on the use case and a reasonable benchmark. Is it better than nothing, better than next best alternative? And how severe are the consequences of making a bad prediction? When is a bad prediction better or worse than no prediction? We've got a comment coming through the chat from Mitzi Morris, who, for those who aren't part of the Stan community, is one of the wonderful developers who makes Stan as great as it is. And she's asking in particular about the ML ops. How do you validate that the model in production continues to be the appropriate model for the data? For example, if the assumptions you made about the data range were too conservative.
Yeah, that's a great question. And that broadly falls under what we think of as performance monitoring. So if you've deployed a model and you're using it to make predictions, for instance, to like unsampled areas in space, you want to make sure that those predictions make sense, right? And a lot of this comes back to where the original data come from. So if you have like a really good sampling design, you're confident that the data that you've collected are capturing the population of interest, then that's a good starting point. But there are some really specific things that you can check in production, just as like sanity checks. So for instance, one thing we check is whether the amount of basal area, which is like the cross-sectional area of a tree trunk, if you sum up the amount of basal area in a pixel, and you're predicting that there's more basal area than there is pixel area, that's physically impossible. And that is like an indication that something has gone wrong, right, with your model. You're making these nonsensical predictions. Another one would be knowing when you are extrapolating. So if you have predictors in your model, like let's say you're using climate principal components, and you're making predictions for a pixel whose principal components are like way outside of the range of observed data that you used in training, that's something you also want to flag. So just knowing when you're extrapolating can be really helpful. So you can then look and see like, do those predictions actually make sense with what we know about the system? Yeah, so those are two specific examples. The crux is when you're just generating predictions from these models, you don't have the right answer. You don't have the target, you have the response variable. You're just like making predictions given a set of inputs. And so you are in some ways reliant on those like sanity checks, and reliant on having done a good job building a trustworthy model to begin with. One last thing we do here is we have test data, external data, where we actually do know the right answer. And that's one more thing that we use in production. Like, these models have never seen this data before. If we're going to deploy it, see how we do on that test data. And that gives sort of one final check to make sure that the predictions are good.
Question for Era. As we get towards these more and more complex models, or models with separate process and observation components and so forth, if we really go down this route, does there come a point where we just sort of lose the stakeholders, where we can't hold their hands any further, and
very quickly.
And is there a solution, or other than simplifying our model, or sort of going over our stakeholders' heads?
We're actually in the process of that now. I sit on the Middle Rio Grande Endangered Species Collaborative Program on their Science and Adaptive Management Committee. So we're like wrangling science and projects to come through. And one of the things that we often hear from managers is like, well, it's great we have this model, but I don't understand it. And I'm not sure what's actionable out of it. And this was doing like population estimates for minnows in the river. And it gets complicated very fast. So one of the things that I suggested is that strip the model back to basics and then take it through a point where it's as simple as possible without, but still giving somewhat realistic answers, right? So like, how do we, because the managers need to be able to understand that the model was built from that iterative process. So the end model, like, we can go crazy in depth to the end model, but you want your stakeholders along the way, because you may just be starting with a simple like linear model and showing, oh, well, this doesn't actually capture the system, right? And we know that. So having that, but having it stepwise, having your stakeholders check in with that constantly, that helps when you actually need to go to these really complicated sort of process models. And that's the best way I've found, is when you get new managers coming in, you start the conversation all over again now. And that can be tiring, but it's, in the long run, it's worthwhile if you want to maintain those stakeholders.
Question for Harold. What were sort of some of the core results or stories that you were after out of the model that you presented?
Yeah, so the core results that I was after is in the kind of downstream analyses, where I was looking at trying to look at the meta community processes that were underlying the patterns that I was seeing. And so I was looking at association between various traits and the habitats and regions where these species were, and using various kind of matrix partial derivatives to show the relation between those, and to be able to show that actually these perennial polycultures, which are kind of a novel farming ecosystem, are likely actually supporting kind of, or species are there because of competition and because of niche compartmentalization, rather than because of just like they happen to be there because of they flew there, but actually, or randomly, and it's actually not a good habitat. But actually trying to show that they're there for reasons that likely suggest that those habitats are actually supporting those species. And so the downstream analysis was really important for us, and so that's why having the modeling structure from our Bayesian model and being able to look at just variation across the local habitat or across the whole region allowed us to get a lot more accurate understanding of the dynamics.
It's really cool. I actually had another question for you. I noticed you use a reparameterization to fit your models. How difficult was it for you to, like, you're fitting these really complex models wonderful data sets, but really like going through the process of which parameters to parameterize, like how did you learn that? I guess.
Yeah, so at first I didn't have anything parameterized, and I was getting all these divergent transitions in the wrong part of the pairs plot, and suggest that they're kind of not just a adapt delta kind of fix. And so this was on a simpler model where I only had a couple covariates. So first I reparameterized those, fit the model, it worked, and then added some more variables. It didn't work, reparameterized those, it worked. And then I think the last couple of variables I added those already reparameterized. And that was working. And for the normal models, it's pretty simple. For some of them, more things in logistic or logit space, it was a bit more complicated. But the Stan resources are really great for those.
I have a question for Era. So you mentioned building priors through dialogue, and other folks have mentioned like building up complexity slowly. And certainly I have felt in the past the compulsion to start with a fancy model. I'm curious whether you have encountered that when you're working with stakeholders who might not be like Stan nerds. Is their inclination to start simple, start with something fancy that incorporates like all of the dynamics and history that they think about, or somewhere in between?
It's usually it's very fancy, like a lot of times, because somebody heard something, they're like, wow, there's this thing called machine learning, we should just throw the data at that. I'm like, no, right? And they're like, want to throw everything in to capture all that processes. And so it's usually on my end to say, hey, let's slow down, because like often, as I point out, I'm not the smartest person in the room when it comes to your system. Like if you're looking at a very particular fish, I know nothing. So I'm not a fishologist, right? It's not what I do. So I start asking lots of questions. That gets to that point about ask leading questions, right? And then that allows me to start with a simple model and eventually build up to that. But yeah, a lot of people want the new shiny, super fancy thing right away, and that's often a disaster on both ends, and it leads to a lot of stress. But having that conversation allows us to kind of step back. Like I couldn't imagine doing what you're doing, Max, with like the tree thing. Like somebody's like, I want this immediately. I'd be like, I would not even know where to start, right? And so it'd have to be something really, really simple. To begin with. I'm curious how like other folks have handled that, because I think most of us interact with stakeholders in some way or another.
All right, well, I'm going to be out of commission for a second as I try to set up this alternative Zoom format real quick, and I'll dump the link over. But carry on.
So I was mentioning the kind of reparameterization that I used. I don't know if there's other kind of tricks that other people have found useful in fitting ecological models that maybe people can share.
All right, well, you dragged me back in, so I'll share one, which was actually, I wish I had time to talk a little bit more about it a little earlier, but one trick that turned out to be really important for us was reparameterizing variance terms. Here, I'm going to share, if I can, just for a moment, if I can find Zoom. Share screen. Share this one. So when we had, for example, random effects of both cluster and subregion, or random effects of both species and family on a term, often the model wasn't too sure about where to put the variance, which level to attribute it to. And so one reparameterization that we found to be really useful was to put the prior on the combined variance of the two terms, and then partition it to the two or more terms using a probability P here, or really this is shorthand for a simplex, right? P defines a two-dimensional simplex, but if we had more nested levels, we could do higher dimensional simplexes as well. And this simplified the prior elicitation, because a lot of times, like, we have a sense of how variable species are in general, but how much of that sits at the family level versus how much of it sits at the species level, or we have a sense of how variable clusters are, but how much of that is really at the cluster level, how much is at the subregion level. So it simplified our prior elicitation in a lot of cases, and it also broke correlations when the model wasn't sure where to put the variance. It leads to these negatively correlated variance terms, but we can reparameterize with a single variance term and then an uncorrelated simplex that tells the model where the variance goes. So that was really useful for us.
That seems like an awesome trick, Jacob. I don't know if there's I don't know, you have a bunch of these things written up. I don't know if that one is written up, but I don't know if there's a way of putting some kind of a central link area on the Stan website or something that links to a lot of these great resources that people have, these tricks that are often kind of hidden around various people's GitHubs, but trying to make a central way of people accessing them. I don't know if there's that's something that, I don't know, Stan can facilitate. Maybe Max for the comments on that, but.
And I'll just mention quickly, I'm not sure if Mitzi is still in the meeting or not, but it's actually a trick that I got from her amazing write-up of some of the conditional autoregressive model parameterizations in Stan, where it's used when spatial versus non-spatial variances are difficult to identify from one another. You put a single prior on the total variation and partition it this way. So thanks, Mitzi, for those write-ups, which have been super helpful to me.
I'll kind of jump in. So back in the day, pre-pandemic times, I had this idea to do the same, do what you mentioned, of kind of putting everything together into like a Stan Ecology website. And so I launched, so I put together Stan Ecology, I put the link in there, the sort of Stan Ecology GitHub page, in hopes that we can all come together as a community, put resources there that have been helpful. I think I've like launched it the first week of March 2020. And then, well, yeah, exactly. Then we all know what happened. So then life got a bit more difficult for everybody, and it just didn't seem like it was something that it didn't need our attention right away. But I'm hopeful that sort of moving forward, that a lot of us can take charge. We can, a lot of us here, can revamp the website or include links, and I want to contribute and continue to grow it as a community. So.
That's terrific. I don't know if, maybe not right now, but I don't know if there's a process for kind of getting things on there or sending things to you, or if there's a group of people that are in charge of running it, or.
No, so far I think I've chatted with Maxwell. It's just kind of me on the GitHub page, but yeah, I think maybe we could start a chat on the Discourse or in the next the breakout rooms. And yeah, the idea is that it shouldn't just be me. It should it should really be like a community ownership type of thing where we can just share resources with each other, and especially help each other with those reparameterizations, or I found this really helpful, some code. And I want to contribute my side from the HRM side as well. So yeah, hopefully it can be useful.
And I'm still working on getting these breakout rooms set up. Just give me 1 second and it should be ready.
Fair to say that pull requests are welcome on that Stan Ecology website? Okay, cool. I just pasted the link in the chat. Yeah, I'm curious. We're all Stan Ecology, at least adjacent people here. I wonder what else we can do to like lower the barrier of entry to this community and make it easier for people to onboard and start contributing.
I don't know. I think so for instance, I'm teaching an ecological stats class next semester, and they don't know it yet, but we're using Stan, and I'm going to kind of go through and, try to prioritize making resources that are useful, especially for newcomers to Stan, what's useful to know about Stan for ecological problems, what's accessible. But I think a lot of us, we already, when we were starting to learn Stan, we always go through these like simple, here's these simple models, here's my prior. I think we all have those files on our laptops or desktops somewhere. So it'd be nice, just if all of us just contribute like one thing. If there's just one thing you can contribute. Maybe it's a little beginner side. I'm not sure. And like I said, that's why it shouldn't just be me. I definitely don't have all the answers, and I shouldn't be driving Stan Ecology. And so I'm hopeful that others have much better ideas than I do, and I'm willing to put in the work and help out.
All right,
And as
I'm gonna stay open over here in case this doesn't work or there are problems or people have questions, but I've got 4 breakout rooms are up and running at the Zoom link that I just put in the chat. So if anybody wants to move over to those, you will be able to self-sort, or I think you'll be able to self-sort into the rooms, which and if you're not able to join for some reason or need a passcode or something, which I tried to disable, let me know.
Mhm.
I seem to have some trouble getting into that link.
So Jacob, it looks like we did need a password.
Cool, I will get you a password. Some people are making, sorry, I have to mute over here because now I have 2 Zoom sessions running. Some people are making it in without a password, and some people seem to need a password. So I am going to find the password and give it to you. Give me a second.
Because I lost the link when I got booted out of here. So.
There's a strong selection bias, but okay, I'm going. How do you join or see the rooms even? Oh,
Okay, so I think that Max Joseph should have made it in by now, I want to say. And maybe it's like you and me.
Oh, yeah.
So let me hang on one second. We do hear you, Max. What ended up being the secret to get in?
I tried again. I just followed, Yanni just shared a link on the chat and I just followed that. I think that one had the password embedded or something. I don't know.
So, Era, I'm hearing from the other meeting that just like clicking the link multiple times, or maybe might help.
Right, could you repost that link for me, please? I ended up losing it.
That one seemed to work for me at least.
Sorry, Harold. Which one seemed to work for you?
I just shared it in the chat.
Okay.
That's the one that Yanni left on the Discourse thread.
Okay, on the Discourse thread.
Yeah.
All right. Hang on.
No worries. Thanks for the help.
So if you send that link maybe out to the event, right?
Yeah, will do. I just have to get the link from the. I'm on 2 computers right now to make the multiple Zooms work, so I have to get it from one computer to the other.
Yeah.
All right, so Era and anyone else who's still here, we have a link that I need to track down that apparently works. So I just need to find
Harold, yeah. Yeah, it's a
that.
Danish pronunciation.
Oh, it's a Danish
Yeah.
pronunciation?
Yes.
Am I right in thinking that like once you've got a multi-species English and mixture model?
Yeah, exactly.
Has that been like computationally challenging at all? I'm assuming you had to marginalize over here.
Yeah. Yeah, it was definitely computationally
Oh,
challenging.
that's not working. Hang on.
And I
Here
don't
we
know,
go.
I tried vectorizing some things that made it not run. So things aren't, it's mostly for loops. There's some things that are vectorized, but I'm not sure why. But
Sorry, Era, I'm almost here, I think.
yeah, it's a bit.
Okay, try this link. I don't know if it's the same link or a different link. It looks like it's the same link.
Figure out kind of what the next steps are on getting it to be a bit more flexible.
How long is you still think it's a while?
Not that like 2.5 days. Manageable.
Harold, do you have visit specific detection covariates or?
No.
Yeah.
That's why I think basically all the visits were basically they were controlled in the observation phase. They didn't really be statistically controlled for. So I felt like I could just ignore the visit specific covariates.